gives me great pleasure to introduce Professor Kathryn North, who is the director of the Murdoch Children's Research Institute and the David Danks Professor of Child Health Research at the University of Melbourne. She is also on the Scientific Advisory Board at Precision Healthcare Singapore, PRECISE. She was appointed as a co-chair of the Global Alliance for Genomics and Health. In 2014, a collaborative network of over 500 organizations across over 100 countries funded by the NIH and the Wellcome Trust. Commencing in 2016, she leads an NHMRC-funded national network of over 80 institutions, the Australian Genomics Health Alliance, AGHA, we have to have an acronym, to provide evidence and practical strategies for the implementation of genomic medicine in the Australian health system. Professor North.
I love the music as you come up onto the podium. It's very Oprah. Good morning, everyone. It's great to be here, and I must commend the PRECISE team and the IHCC on organising a fantastic conference. It's a real honour to be here. Now, we've heard a lot, and yesterday particularly from Genomics England and the PRECISE initiative in Singapore, around national initiatives and the challenges around implementing genomics into clinical practice. And so today I thought I'd take a high-level view of some learnings that we've had over the years, both nationally and internationally. So this is familiar to all of you. The Holy Grail is that we have a genome and information that can be used about an individual throughout their lifetime, to diagnose, but to predict and to prevent disease. Now, this isn't as easy as it sounds, because to implement genomics into healthcare requires a revolution in our thinking. And so much of our health systems is focused much more on waiting for disease to occur and then treating it, which is inefficient and often ineffective, rather than thinking forwardly how we can actually prevent disease and the economic models around getting that done. So, as I was thinking about this talk, and also what's happened in my own country, and Australia is very different to England. I dream of having one NHS to rule them all, because you can be cohesive and you can have big ideas. But in our country, we're a federated system. We have a number of different states, each of which have their own governments, and the states run the hospital systems. And we have a federal government that runs all the primary health care, but also pays for medicines, and we have a Medicare scheme that provides a national support for people in health. So it's a hybrid system, but as you can imagine, there's always arguments about who's going to pay for what. So as we've built Australian genomics, and then I think it's relevant to what Heidi was talking about yesterday with the Global Alliance for Genomics and Health, is that I'm going to go through what I think are the big principles that all of you in your countries should be thinking about to achieve that large-scale delivery of genomics. First of all, the strong, and I'm going to give you examples of this, but strong collaborative networks that are multidisciplinary and bring research and clinical care together. Wide geographical reach. Australia is a big country with lots of rural and remote areas. But how do we maximise what we do? How do we take our research and our clinical care throughout that large country and ensure equity of access, which is incredibly important to governments? And an engaged leadership. And, starting from your health minister, I mean, when Sue said yesterday that the Minister for Health, the new Minister for Health, was embracing genomics in his second week, that's what you need. You need to be engaging and working with government, but also with the leadership across the organisations around your country. So I'm going to talk about 3 different examples. Australian genomics, obviously, how we're also putting that in practice through our work with the Global Alliance and the National Initiatives Forum that Heidi mentioned yesterday. And then through what is a driver project for the Global Alliance, which is looking at how we solve some data sharing problems on an international scale, IPCHIP. So Australian genomics came about after a series of consultations around the country, led by Warwick Anderson, who at that stage was the head of our National Health and Medical Research Council. And it resulted in this targeted call for research for $25 million over 5 years, which sounded like a lot to us. It was the second largest grant that had been ever given by our NHMRC, but it sort of pales into significance in some of the other investments across other countries. And it was called Preparing Australia for Genomic Medicine. And this is basically a combination of a health services research and implementation science program, because the goals were to demonstrate how patient benefit can be maximised using genomics, so really the clinical utility, and it said in one or more diseases, building the evidence for cost effectiveness, for implementing it into healthcare, practical strategies to achieve that, but also at the same time building the research capacity around Australia. Now, we had that $5 million a year, but we also had other sources, and I would say we leveraged over that time over $100 million of investment, either in research. Our government announced in 2017 a $500 million investment in genomics research over 10 years, so we could support and work with that. And as well, the state governments were also investing in genomics in their healthcare systems. So we were leveraging a lot of investment, but the goal was to bring it all together in these collaborative networks with engaged leaderships. The approach we took is in this particular diagram on the side. And we're like the opposite of Genomics England. We didn't do a big genomic sequencing project. We started with these flagships. We started within healthcare with what existed and then worked out how we were going to build upon it. Everything we did was prospective, starting with patients as they came into clinics around the country. And we established collaborative networks of experts in different areas around the countries. So obviously starting with the clinical geneticists, but then with the neurologists, the cardiologists, the familial cancer clinics, etc. And we really built then the infrastructure that was needed to take that through clinical practice. And the programs that you see, I mean, there's the All Necessary Diagnostic and Research Network, but at the same time, we built, bit by bit, the national approach to data federation and analysis, the expertise in health evaluation, health economics and implementation science, but also the creation of a genomic literate workforce at all levels within the health system. So this is actually Australian Genomics now encompasses over 100 institutions around our country, so the broad collaborative networks. It also includes every clinical and diagnostic laboratory for genetics around the country, as well as you can see down the side, we plugged into national infrastructure that existed. We have our wonderful international partners, and particularly through the Global Alliance. So we were always concentrating on looking outwards and incorporating best practice into everything we do. We also, very importantly, I think one of our most important committees early on was what we called the National Implementation Committee, which actually brought together representatives from the state governments and also from the major federal agencies involved in funding health, MSAC and the Pharmaceutical Benefits Scheme. So we were having these discussions along the way to make sure what we were doing was going to be thought about at the government level. So, quick high-level summary. This is published the first 5 years. This is a 5-year program of work. And we can see down the slide the flagships. We ended up with 20 different disease areas where we funded, they're like the clinical implementation projects that have been so successful in Singapore, where we brought together the collaborative networks and prospectively looked at usually a couple of hundred patients in each area to demonstrate the effect of genomics on clinical utility, diagnostic rates, and importantly, how it influenced clinical care. I'll touch on the Mackenzie's mission, which Martin Delaticchi talked about yesterday, around reproductive carrier screening. And we then evolved what we did related to what was available. So we're starting with sometimes panels and exome sequencing, then we moved on to whole genome sequencing, and in our latest studies have also added in transcriptomics. So this, I think, is the first big KPI that I wanted to see from this program of work, is that we provided to government the evidence to implement and pay for this in clinical practice. And I'll use from a very early study, the 2020, these are Medicare items, which mean now that every child and every family around the country has access to genomic testing for these indications. And so say the 2020, that's the year of syndromic, non-syndromic intellectual disability and associated childhood syndromes, we demonstrated as part of our pilot, we just looked at standard of care in 100 kids, and in the same 100 kids, we then at that stage did an exome to see what happened. And we demonstrated we could lift the diagnostic rate from 11% to 55%, and the cost per diagnosis to the health system went from $26,000 to $6,000. 5 times the diagnostic rate, quarter of the cost, no-brainer. 3 years later, it became a Medicare item. And I'm going to talk about what's on the left in terms of ones that are currently under consideration, because they need different models of funding. The first is Mackenzie's mission, which was a passion of our health minister, again, that engagement of the leadership. And this was a $20 million project. You know, this is additional funding in terms to our core funding. And we wanted to take genomics into reproductive carrier screening. And as I said, Martin Delaticki was one of the leads. It was a national program of work. And the goals were really also to show that we could do this around the country with equity of access. So what you see dotted around the map of Australia is all the areas where we recruited patients. And we did this study during COVID. Overall, we recruited over 9,000 couples. We did exome sequencing and then analysed panels for specific genes where our experts had said there were indications in terms of your parents would want to know about this to help their reproductive decision making. 1 in 50 couples were identified as being at risk of an autosomal recessive or X-linked recessive disorder. But interestingly, we currently have funded in our country triple testing for Fragile X, spinal muscular atrophy, and CF. But 80% of the disorders we detected were not those 3. So we're now using that information to work with government of how do you take that to scale to the 300,000 births that we have every year. And that's the challenge. And that's also something that is a challenge for all of you that we're really looking at the National Initiatives Forum of the Global Alliance. So the other one was acute care genomics, led by Zanitza Stark and Sebastian Lunk, which was how we could take within our diagnostic laboratory, and luckily my institute also has the Victorian Clinical Genetics Service as a wholly owned subsidiary, so we can translate very quickly into practice. But getting it down from initially 4 months to 4 weeks to 2 weeks to 3-day turnaround time, and demonstrating we could do it and that it was worth it, because it's very costly, because you can't batch samples when you're doing it that quickly, and you have to do trios. So we ended up, led by Zanitza, doing a national study that involved every paediatric and neonatal intensive care around the country. It was prospective. All of the intensive care physicians were investigators on the study, and that's the other way to increase the genomic literacy of your workforce, get them involved in a study, and then get them excited about it, and then demonstrate that it works. And so with the 450 infants that we enrolled over about an 18-month period, we demonstrated that we could provide a diagnosis while they were in intensive care with a 3-day turnaround time in over 50% of the kids. It influenced care in 75% of cases, and it was highly cost effective just looking at savings on ICU beds, let alone what we could prevent. So it's been highly impactful. Our Martin Delaticki, who spoke yesterday, said it's sort of like crack cocaine for the intensive care physicians because they have this tool that they can use that's really making a difference. And so we've also incorporated, and this paper came out last year, using transcriptomics to continually increase the diagnostic rate and make it part of our clinical pathway. But this is in a hospital system, so it's not something our federal government is going to fund. So what I learned is I started to have to start to advocate 2 years in advance. This is a no-brainer to fund, but we had to work with the state. We worked with, got it ticked off by Health Technology Assessment, by our Medical Services Advisory Committee, but then we had to go, and they said, well, there's going to be a delay of a couple of years for the federal government, the states, to make an agreement of how this gets funded. So you're going to have to go and talk to the states directly. And I'm very happy to say that in Victoria, the day that the research study finished, they agreed to fund it. So it's now standard of care within Victoria, within Western Australia. Some of the other states send samples to us to do it because there's an economy of scale here as well. But we're also working with government to find what's the funding model that can make this work, because it's obviously worth it. So after all that great work and a very engaged health minister, by November 2019, it became obvious that you need to bring this sort of work not as a bottom-up research program that's engaging upwards, but to have a top-down approach with an authorising environment and the authority and the leadership to make this happen. So in November 2019, our health minister convinced Treasury that Genomics Australia should become an entity and a government agency. Then there were a lot of bushfires in Australia, and then COVID happened. And so it wasn't the time to set up a new agency in the country. So what they did is they funded us initially for the next 3 years. This is a very long story of interacting with government, but not to be doing the research ourselves, but with the really expert team that we had, led by Tiffany Bortwood, who's somewhere in the audience, there you go, that is really bringing this expertise as a connector and coordinating genomics around the country. So we still support research programs, but we're engaging across the health system with the delivery services, with government. We're making sure that research studies are providing the evidence that can be translated into policy and then into practice and have that health economics part about it. We were also authorised, which is very important, by the Minister and the Head of the Department of Health, to do a business continuity plan and a national consultation just on what genomics in Australia should look like going forward, and then to prototype a national approach to genomics information, where a blueprint had been developed by government, and then we prototyped it with a sort of attended call around the country. And there's a number of people in this room that also provided expert international advice on what that would look like. And we also provide points of engagement with industry. We work very closely with our Indigenous community, and obviously we work very closely with global partners such as the Global Alliance and WHO. So the scope in terms of the research that we support has grown. You'll see in a number of these research projects, it's not about specific disease areas, but it's encompassing the sort of national acute care genomics, looking at how we implement new technologies such as automated reanalysis and are doing that on a national scale. We also have an Australian Functional Genomics Network, which is really looking at how we improve the interpretation of VUS and enable gene discovery. And it's very much modelled on the Canadian system that's been running for many years, but also there are links across Japan, Canada, into an international functional genomics network. We've also, over the years, developed digital infrastructure that's supporting the integration of genomics into practice. So Mackenzie's mission was done completely, the reproductive carrier screening, using a dynamic consent platform called Control, which is, again, all of our work is open source and ready for adoption and use. We worked with Genomics England and particularly Augusto Rendon in implementing an Australian form of panel app, which looks at gene disease associations continually updated. And also that's been, Zanitza has run lots of workshops, and this is probably the most used thing on our website. It gets something like 150,000 hits a month from over 50 countries. We've developed Sharient, which is my favourite name for a digital tool, and that's actually a national platform that links all of our diagnostic genomics laboratories so that they can internally share the variants that they find and the levels of evidence. And then we've built, working with Heidi and the team in ClinVar, we've worked, and Global Alliance has built an API so that everything that comes out of our diagnostic labs can immediately be uploaded with the levels of evidence to ClinVar. And then, as I've said, we did the prototype of implementation for the national approach to genomics information management. And then there's involving the community across a whole different number of areas. And again, learning from our our overseas colleagues. So Involve Australia and the guidelines for engaging consumers into research and genomics is very much based on the work from Genomics England. But just as down the bottom, all of the tools and resources that I'm talking about are open source and available on our website. So we still don't have Genomics Australia, a government agency, and the reason was that actually the very enthusiastic health minister in March 2022 basically made the announcement about the formation of Genomics Australia and that it would become a government agency, and announced a forward budget for that, and that it would also be in partnership with our state governments with co-contribution. And then 2 months later, that government lost the election, and then we had a whole new government. Luckily, I had pre-briefed the then Shadow Minister for Health. But that government decided they wanted to wait for 6 months before they issued a new budget to decide whether things would get funded that were promised by the previous government. Luckily, Genomics got through that. And then with the DepSecs and the government departments we'd been working with over the previous 5 years, they did a government restructure, so we lost all of those people and got moved into a new area. And then we started again. I'm just telling this story to say, it's not, this can look glossy and great, but it's not easy in the background, and that relationships that you're having with government are so important, and the continuity is so important. But we're getting there, and Genomics Australia is basically Australian Genomics. They wanted to change the name around so it sounded like it was different as a government agency, but it's really all the things that I've talked about is what Genomics Australia will do going forward, with a model, hopefully, that hasn't been negotiated yet, of co-contributions from the states. And we've had an expert advisory group that I've co-chaired with government that's reviewed what the functions and priorities will be. And as I said, the goal is that this will be up and running in July 1st next year. We'll see. So I just want to spend the last 10 minutes talking about other areas that I think are incredibly important. You know the Global Alliance, and I've been involved in the Global Alliance almost as long as Peter Goodhand, since 2013. But this is about how we come together as a large global community and network to develop standings and frame policy for sharing, responsibly sharing genomics and healthcare data. The reach continues, and it's so wonderful having this conference in our Global Alliance conference next month within the Asia Pacific. And it's so, again, this is just stressing the importance of collaboration over so many countries so that we have the standards when we want to share data, and we're promoting the sharing of data and looking at how we create the resources to do that. And Heidi's talk yesterday just beautifully demonstrated the sort of collaborative work that needs to happen for us to be able to make all of this quicker, faster, and as accurate as possible. So again, you know, the circles of going from the patient and also reutilisation of information for research. This is the learning system that underpins the Global Alliance, and as I've demonstrated, certainly underpins the way we've developed what we do in Australia. And over this past 10 years, you can see that all the way along the pipeline, the Global Alliance for Genomics and Health has developed the standards to promote that sharing and interoperability of the data that we have. So I think that, and again, all of this is open access, available in the pre-competitive space for everybody to use. We then started grappling, as we had all of these tools, to think about, well, how do we implement this at scale? It's no use having a lovely website with lots of stuff on it that people can use if we're not promoting how it gets used more widely. And so actually in 2016, it was then Mark Caulfield from Genomics England and us in Australia and the Global Alliance decided to start convening what were at that stage often nascent national initiatives to bring together to look at opportunities for collaboration, to facilitate and really encourage the use of standards at scale, to promote sharing of data and its use in healthcare. So from the national initiatives, there is around large-scale initiatives that are promoting genomics into healthcare. But also it ended up being a bit of a counselling session. We'd learn from each other's problems and help each other. And really then we're at a stage now, and this is the current membership. You go on the Global Alliance website, there's a simple form. It's a great community that's well established and is building. And I think this is what we're aiming to do, is really make sure all of our data is interoperable. Heidi talked about the variant sharing yesterday, and as well as developing the network of nomads, because that's the quickest way that we can really increase the diversity within our databases, is by linking and using all of these tools and contributing to them at the same time. Then the last thing, just to show that this sounds great, but it's not easy. IPCHIP, the International Precision Child Health Partnership, is a driver project for the Global Alliance. And this is sort of in my day job running my Children's Research Institute. I work quite closely with Boston Children's, with Great Ormond Street Institute for Child Health, and Toronto Sick Kids. And we all know what we're doing in genomics, and we're all early adopters. We've all got sort of quite well-developed data systems. We all collaborate. But we thought we'd like to bring together those groups because we can much more quickly do these prospective studies where we can bring together groups of kids with rare disease, diagnose them, how to promote their diagnosis, and get them into clinical trials. How can we start to do that at scale? So it's really around sharing our knowledge, but our goal is how do we improve the outcomes of kids with rare disease? And it's because, if you see that bottom right-hand corner for you, is there's only 12% of disease-associated genes that have an established intervention. And that, from a paediatric perspective, is where we want to start lifting that. We've got some great things like the treatment for spinal muscular atrophy, cystic fibrosis, but how do we expand that? So that's the whole point of IPCHIP, is how do we start to take that at scale? But what we've found is it's really hard. We wanted to share our genomic databases, and let alone sort of getting over the regulatory requirements. What is the platform we can use to actually share data on a genome scale and just not on a variant scale, linked with machine-readable clinical data? And so that's what our driver project is doing with the Global Alliance, because I figure if the 4 of our institutions can't work out a way to do this, then that sort of idea of easy data sharing at scale is not going to happen. So that's what the driver projects in the Global Alliance are aiming to do, is work out where those barriers are and not just create a system where we all create one database and all have the same platform. It's about how do we increase interoperability at that stage, to not just share variants, but to be able to share genomic data. And so I thought I'd finish with what's been our first pilot project with IPCHIP, because it all comes back, really, although I'm fascinated by the process and how we make this really broadly available, it all comes back to what we can do better for our patients. And so in deciding on what we'd start with, we wanted to do something where we knew there was, very large numbers of genes associated with epilepsy, that it's a disorder that's, 1 in 1,000, but between our 4 institutions, we reckon we could recruit around 500 patients per year. The problem with the treatment, these need targeted treatments. These kids, if they keep fitting, also become very intellectually retarded, developmentally delayed. And so it's a high healthcare need, genetic cause in at least 50%, probably more. But genomic testing and diagnosis and trying to find a specific diagnosis either doesn't happen or takes months, by which time it's too late. And so we've just recently published the pilot study, but this is on the first 100 patients. But we found that working together on this, we could accumulate this 100 patients in less than a year. It had a 43% diagnostic yield. We were aiming initially for a 2-week turnaround, but we're bringing that back, so we're taking it to the really rapid genomics, around a 5-day turnaround. But then looking at how can we change the trajectory of these kids in terms of their seizures and their development. And this is continuing. I think there's currently around 300 or 400 kids that are enrolled in this study. And just to bring it to life, is in my final slide this is Levi, who presented on the first day of life with very severe seizures, and started on the usual, but to no avail. Had to be intubated, he had to be tube fed, and so the prognosis was very poor. We enrolled him in the study, and within 3 days, he had the diagnosis genetically, for which there is a targeted therapy. So get rid of the phenobarb and the phenytoin. And this is Levi at 1 year. His seizures stopped. He's still have some developmental problems, but his developmental trajectory continued. And actually, he's now 3 years of age. He and I went and met the Wiggles together, which I don't know if you know the Wiggles, but they're very excited. I was quite excited, I must say. And he showed me his kung fu moves. So, this is treatments that change lives, and this is why it's important. And if you want to excite your leadership, you show them these sort of stories and get your patients to advocate. So I hope I've brought that together of principles you should be thinking about. It's in common no matter which system that we work with, in terms of the networks, the reach and the engagement of leadership. And as we're thinking this through, we're actually putting together just an opinion piece on this that we'll publish, and very happy to share, around using some of our examples about how these networks, that making sure you've got wide geographical reach and engaging leadership at all levels. And plus, probably add in here, how do you work across so many multidisciplinary groups that have different focuses, but also make sure you can all speak the same language in terms of all the people that need to be involved to make this successful. So we've got our plenary for the Global Alliance coming up next month. We've got Australian animals coming to the welcome reception. And just my team on the ground, I've already mentioned Tiff. I haven't got a pointer. She's there. John Christodoulou, that many of you know, is working with me on the IPCHIP program. Zanitza and Seb up in that corner, many of you will know, who've run our rapid genomics program. So thank you very much.
Thank you, Professor North.